Xin được chào mừng toàn thể các quý vị đại biểu đã tham gia buổi hội thảo trực tuyến tăng cường khả năng phát hiện biến chủng Omicron bằng xét nghiệm sinh học phân tử của chúng tôi ngày hôm nay. Cảm ơn quý vị đã dành thời gian tham gia của chúng tôi. Chương trình hội thảo sẽ bắt đầu sau 5 phút.
Kết hôn. Hospitals are on high alert for the deadly coronavirus.
Studies finding elderly are most at risk.
Coronavirus resurgence threatens economic
The
recovery.
lockdown has been expanded.
In early March 2020, the World Health Organization declared COVID-19 a pandemic. As more and more people became ill and economies began to shut down, clinicians and scientists started working together to find answers. It became vitally important to develop a diagnostic test, which is precisely what the scientists at Thermo Fisher Scientific did in record time. Our real-time PCR test, which diagnoses the virus in less than 3 hours, was authorized by the United States FDA for emergency use and later received the CE mark in Europe. The Applied Biosystems taqpath COVID-19 test kit is now available in nearly 180 countries. Why was this kit so rapidly adopted for use around the globe? Because regulatory bodies throughout the world know what we all know, that to combat the pandemic, we need accurate, proven, gold standard technology. We need to work on a global scale. We need scientists to accelerate innovation with urgency. And that's why Thermo Fisher Scientific is leading the world in COVID-19 testing. Our scale and depth of capabilities have never been more vital. And our mission to enable our customers to make the world healthier, cleaner, and safer has never been more important than it is today. From the beginning, Thermo Fisher mobilized supply chains, activated R&D resources, and deployed field teams to enable our customers to meet the demand for COVID-19 testing all over the globe. From New York to Los Angeles, Sydney to New Delhi, Rome to Frankfurt. We know how important it is for you to have access to reliable supplies for COVID-19 testing. And it's our top priority to support you during these challenging times. Even as parts of the world continue to shut down and face other COVID-19 challenges, we deploy local support teams to keep your labs running, anytime, anywhere. When on-site visits are not ideal, our service and support teams are there to guide labs remotely. With our industry-leading supply chain and world-class service and support, Thermo Fisher is leading the world in COVID-19 testing. 50% of COVID-19 testing is performed on Thermo Fisher technology, and we have the capacity to manufacture tens of millions of tests per week, making us your trusted supplier for your ongoing COVID-19 testing needs. Get your reliable supply of COVID-19 test kits today at thermofisher.com/COVID-19.
Công ty cổ phần thiết bị SISC Việt Nam xin gửi lời chào và lời cảm ơn chân thành tới sự quan tâm và hợp tác của quý cơ quan trong thời gian vừa qua. Công ty cổ phần thiết bị SISC Việt Nam là đại diện phân phối chính thức tại Việt Nam của hãng Ion Torrent và Applied Biosystems của Thermo Scientific Mỹ tại Việt Nam. Kính thưa quý vị đại biểu, đại dịch COVID-19 đang ngày càng diễn biến phức tạp. Virus SARS-CoV-2 liên tục xuất hiện các biến chủng mới, gây khó khăn trong công tác phòng chống dịch bệnh. Ngày 26 tháng 11 năm 2021, WHO đã xác định biến chủng B11529 hay Omicron là biến chủng đáng lo ngại. Đây là biến chủng có số lượng lớn các đột biến, trong số đó nhiều đột biến
Xem một cái training của hãng
có thể liên quan đến khả năng lây truyền và khả năng trốn thoát hệ miễn dịch. Đây là vấn đề mà tất cả các quốc gia trên thế giới rất quan ngại. Vì vậy có nhiều quốc gia đã thắt chặt việc nhập cảnh nhằm kiểm soát hạn chế sự lây lan của biến chủng này, trong đó có cả Việt Nam. Trước đây, các ca nhiễm biến chủng Omicron tại Việt Nam đều là trường hợp nhập cảnh được cách ly. Tuy nhiên vào thời điểm tối ngày hôm qua, chúng ta đã ghi nhận 3 ca nhiễm trong cộng đồng đầu tiên. Những trường hợp này được xét nghiệm real-time PCR cho COVID-19 tại Bệnh viện 30 tháng 4. Các anh chị kỹ thuật viên tại bệnh viện đã sớm cập nhật thông tin về hiện tượng gen S không biểu hiện trong xét nghiệm COVID-19, có thể là biến chủng Omicron. Do đó, khi thực hiện xét nghiệm bằng bộ kit taqpath CE-IVD của Thermo Fisher, 3 mẫu này có tín hiệu gen N và gen ORF1, nhưng lại không có tín hiệu ở gen S. Nhận thấy sự bất thường của những mẫu này, các anh chị kỹ thuật viên đã kịp thời báo cáo và các mẫu này sau đó đã được gửi tới Bệnh viện nhiệt đới để giải trình tự và xác định đó là biến chủng Omicron. Việc lây nhiễm biến chủng Omicron trong cộng đồng là một vấn đề rất đáng quan ngại. Nhờ có sự cảnh giác cao của các cán bộ kỹ thuật viên tại Bệnh viện 30 tháng 4, chúng ta đã sớm phát hiện các ca nhiễm Omicron để kịp thời có những biện pháp khoanh vùng và giám sát. Với mong muốn trao đổi, cập nhật thông tin, các giải pháp và tăng cường khả năng phát hiện biến chủng Omicron để sẵn sàng ứng phó với dịch bệnh COVID-19, chúng tôi kết hợp với hãng sản xuất Thermo Fisher Scientific tổ chức buổi hội thảo trực tuyến ngày hôm nay. Tăng cường khả năng phát hiện biến chủng Omicron B11529 bằng các xét nghiệm sinh học phân tử. Sau đây tôi xin giới thiệu đại diện đến từ hãng Thermo Fisher Scientific là ông Christopher Ong. With a desire to exchange and update information on solutions to enhance the detections of Omicron variants, we are ready to respond to COVID-19 global pandemic. Our company, together with the manufacturer Thermo Fisher Scientific, has organized this event today, the online webinar, Enhancing the Capability of Omicron Variants Detections by Molecular Biology Test. Here, I would like to introduce a representative from Thermo Fisher Scientific, Mr. Christopher Ong.
Hi, good afternoon. So first of all, I would like to thank you for joining us this afternoon. I understand that there is still a lot of uncertainty right now about the COVID-19 situation around the world. So we will have to make sure our responses to the pandemic has to be quick enough to keep the public health protected. So that is why I want to thank all of you here, whether you are from the healthcare or diagnostic sector, on your effort in keeping the people safe. Therefore, we are excited to share with you today our full range of COVID-19 testing solution using real-time PCR, Sanger sequencing, as well as next-generation sequencing, NGS, to help you to deal with the latest variant of concern for COVID-19, which is the Omicron variant. I hope you will find today's session informative and useful, helping you to prepare for the transition towards living with COVID-19. With that, I thank you for your attendance. Back to you, Lan Anh.
Thank you very much, Christopher. Nội dung chương trình hội thảo hôm nay bao gồm phần trình bày của đại diện hãng Thermo Fisher về các giải pháp sinh học phân tử giúp tăng cường hỗ trợ phát hiện và nghiên cứu biến chủng Omicron, trong đó bao gồm giải pháp real-time PCR, giải pháp điện di mao quản hay giải trình tự Sanger và cuối cùng là giải pháp giải trình tự gen thế hệ mới NGS. Tiếp theo đó là phần thảo luận được dẫn dắt bởi tiến sĩ Phạm Thu Hằng, giám đốc trung tâm CGG Center, và ông Christopher là người đại diện cho hãng Thermo Fisher sẽ giải đáp các câu hỏi của quý vị. Bài thuyết trình đầu tiên, chúng tôi muốn giới thiệu với quý vị trong buổi hội thảo có tựa đề Biến thể Omicron, các thách thức trong việc phát hiện và sàng lọc biến thể SARS-CoV-2 bằng xét nghiệm PCR do ông Christopher trình bày. Để thuận tiện cho việc truyền tải thông tin đến quý vị trong thời lượng ngắn gọn của buổi hội thảo này, chúng tôi xin phép được trình chiếu các bài trình bày mà ông Chris đã thu riêng cho buổi hội thảo ngày hôm nay, kèm phần phụ đề tiếng Việt. Chúng tôi hy vọng cách làm này sẽ giúp quý vị có thể dễ dàng theo dõi nội dung hôm nay hơn.
Good day, ladies and gentlemen. This is Christopher Ng, a regional technical sales from Thermo Fisher Scientific. Just a quick introduction of myself. Prior to joining this company, I served as a medical technologist at Molecular Diagnostics Center, National University Hospital of Singapore. I have more than 7 years of experience on validating and running clinical molecular diagnostic tests, covering infectious diseases, mutational analysis, heritable diseases, as well as leukemia. Given that clinical molecular diagnostics have gained prominence recently, especially during the COVID-19 pandemic, the emergence of new SARS-CoV-2 variants poses new challenges not only for COVID-19 detection, but also variants
Cái này như cái vô
to be here today to share with you some key updates about the Omicron variant and how do we address these concerns through molecular diagnostics using our real-time PCR solutions. Before going into the content, let me first briefly introduce you the company I am representing, Thermo Fisher Scientific. Our mission is, with the wide spectrum of life sciences solutions, we aim to enable our customers to make the world healthier, cleaner, and safer. In terms of our teams, we have established global thought leadership in all areas of science and harnessing the power of all that knowledge. With more than 10,000 customer-facing reps, so we can always be there for our customers. No matter what science, we have focused experts and scientists in-house that are focused on the same things. We are also committed to advancing science and staying ahead of innovation through research and development, generating more than 25 billion of revenue per year. As you can see, our work has been largely business to business, serving the society through our partners. Today, with the COVID-19 pandemic, we have started doing so in a more direct way. Thermo Fisher is at the forefront of the global response to COVID-19, leading the enablement of our partners in the fight against the virus. We support the research of the virus, diagnosis of infection, providing essential lab equipment and personal protection equipment, as well as supporting development and production of new treatments and vaccines. Today, over 50% of COVID-19 testing are performed using the Thermo Fisher technology. When the coronavirus outbreak began in late 2019, Thermo Fisher Scientific rapidly developed one of the first COVID-19 diagnostic tests, which has now scaled to manufacture 20 million tests per week. Thermo Fisher Scientific is working to apply its skill as the world leader in serving science to standardize and industrialize the manufacture of tests and testing platforms in a reliable and reproducible way to support the global need for testing at scale. Globally, over half of all COVID-19 testing is currently being performed on Thermo Fisher Scientific instruments. We have rapidly increased our production of COVID-19 kits and continue to increase our manufacturing capacity to 20 million reactions per week and beyond if needed. Labs have been constrained with supplies from reagents, consumables, and instruments. Thermo Fisher will provide assured supply for your lab to ensure that there are no concerns in your workflow and throughput. For COVID-19 testing, Thermo Fisher Scientific is vertically integrated. We are a single supplier and distributor for consumables, reagents, instruments, software, and 24/7 service and support for labs running or looking to run COVID-19 testing. Our world changed overnight, and we took action. We aspire to developing testing solutions you can trust for the information you need to get the answers and keep moving. As we all aware, viruses mutate, especially RNA viruses like SARS-CoV-2. Why is that so? The uncontrolled spread of COVID-19 globally has contributed to conditions that favors virus evolution. Multiple reports have also showed that the virus has been mutating at the rate of about 1 to 2 mutations per month, especially for some recently detected variants mutated even faster beyond scientists' expectation. What might new SARS-CoV-2 mutations and variants mean? And why should we worry or be concerned about that? These variants might increase the speed of human-to-human transmission, change the severity of the disease, affect the susceptibility to therapeutic agents, evade vaccine-induced immunity, as well as impact detection by clinical tests. Therefore, we have this classification of SARS-CoV-2 variants that helps inform public health, lab, and enhance community response. In terms of variants of interest, They are known to carry genetic changes that affect virus characteristics such as transmissibility, disease severity, immune escape, therapeutic or diagnostic escape, with community transmission with apparent epidemiological impact to suggest an emerging risk to global public health. While variants of concern would meet the criteria of variants of interest, these variants have been demonstrated to intensify the transmissibility or deadly change in COVID-19 epidemiology or decrease in effectiveness of public health. For example, the Lambda and Mu variants have been assigned as variants of interest, while before that, we have the Alpha, Beta, Gamma, Delta as variants of concern, including Omicron being the latest addition to this category. Let us look at some of the responses from public health organizations and government. On November 26, global public health institutes like WHO, Africa CDC, European CDC, as well as US CDC all released official classification and statements about the Omicron variant. Following that, some countries quickly enforced travel restriction or border closures, expediting national vaccination programs. Confirmed Omicron cases were already being reported in more than 20 countries back then. This figure highlights our response to SARS-CoV-2 Omicron variant. If you look at the timeline for Omicron emergence and spread, the variant was first detected in South Africa in late November, followed by a call with CDC 1 week later to discuss about S-gene target failure or S-gene dropout observed from our PCR test. We then launched 2 Omicron-related mutation genotyping assays in the first week of December. There were more than 26,000 cumulative Omicron sequences detected in 81 countries as of December 2021. As we observe further on the Omicron sublineage trends and the 6970 deletion on the S gene of SARS-CoV-2, the BA.1 strain still dominates the Omicron landscape, with 99.5% of Omicron sequences detected. This figure in red also show that the number of samples with S6970 deletion increases steadily over time within just a month. As mentioned, Omicron has spread to 81 countries so far, with most countries becoming the predominant variant. This figure adapted from GIS8.org shows the distribution of Omicron sublineages detected by country, dominated by the dark blue dots or the BA.1 sublineage. There is also a rapid surge in the number of Omicron sequences observed in countries like the UK, Canada, and Australia within just a few weeks of time. With that, I would like to introduce you the taqpath COVID-19 assay, which we designed carefully with mutations in mind. By mapping to more than 55,000 SARS-CoV-2 genomes, we included only the most stable parts of the genome and avoided unstable regions. We identified and targeted unique regions of the SARS-CoV-2 genome to avoid detecting other coronaviruses with 100% homology to all SARS-CoV-2 isolates, primer, probe sequences of open reading frame 1AB, N gene, and the S genes to ensure optimum specificity. We built in redundancies with multiple gene targets to compensate for virus mutation at the primer sites, which has been useful in detecting and understanding both the alpha and beta variants. Of course, now for the Omicron variant as well. This figure simply clarifies how we chose targets in the most stable areas of SARS-CoV-2 genome, with the lower risk of mutation at the time of design. Again, this 3-target design functions to compensate for virus mutations. Let us now discuss further about S-gene advantage and what it means in Omicron variant detection. As mentioned, the B11529 variant, or commonly known as the Omicron variant now, first reported in Botswana and South Africa in November 2021. So far, what we know about this new variant is that it carries a large number of concerning mutations with more than 30 genomic changes. This includes the 6970 deletion on the S gene, as well as 15 mutation in receptor binding domain. The Omicron virus may transmit faster, reduce vaccine effectiveness, and increase risk for reinfection because of these genomic changes. In this case, the WHO published a general guideline that contains recommendations on how we could deal with this new variant through enhanced surveillance, actively report cases, including field investigations and lab assessments. This press release also highlights an important comment from the director of the Center for Epidemic Response and Innovation in South Africa, that Thermo Fisher tests allowed them to detect cases that may contain the Omicron variant through S gene dropout. This early identification will be critical in helping to track and understand the spread of the Omicron variant to South Africa and the world. Attached is the WHO technical brief that indicated Thermo Fisher taqpath assay can be used as proxy for the Omicron variant before sequencing. The S gene target failure approach may lead to faster detection rates. The European CDC also recommended the use of Thermo Fisher assay as a screening method for Omicron. This is yet another urgent briefing that reiterated the utility of S gene target failure feature as a screening tool for the Omicron variant. This was strongly supported by an early evidence that 100% with S gene target failure were the Omicron variant. Furthermore, European CDC also reported that the S gene dropout can signal the presence of 6970 deletion, potentially the Omicron variant, especially in settings that are still dominated with the Delta variant. This approach will quickly help to differentiate patients on whether they are infected with the Delta or the Omicron variant. I would like to just briefly highlight the summary of variants and our test here. We have released and been updating this reference guide to notable SARS-CoV-2 variants based on WHO classification to inform our customers about the latest known impact on our PCR test. As you can see here, that we have included all previously reported variant of interest or concern. In this case, we have indicated the S gene advantage, which means the S gene dropout may signal the presence of 6970 deletion and potentially the Alpha variant, and now the Omicron variant. We will now spend the next few minutes to discuss our research tools available for SARS-CoV-2 mutation and variant surveillance. Epidemiological surveillance is conducted to ensure viral diseases match the reference strain, as well as to monitor possible mutations. This is important as many changes in the viral genome can impact public health policies and options, how the illness spreads in the population, potential study of treatment options, and vaccine development research. There are still many unknowns about SARS-CoV-2 which need to be understood to better predict virus strain evolution. Scientists will need to constantly ask questions like, how can we detect new variants? What is the origin of a new variant and how prevalent it is? Will current molecular or antigen-based tests miss detecting a variant? Or does a variant spread more quickly or lead to higher disease severity? And last but not least, what is vaccine efficacy against a new variant? That is why we aim to become your trusted partner in SARS-CoV-2 surveillance. For strain surveillance, we offer multiple research solutions through next-generation sequencing or NGS, or Sanger sequencing to help sequence the full viral genome or targeted regions of interest. When scientists look to confirm and verify known mutations associated with specific strains or lineages, they could use either our Sanger sequencing or RT-PCR genotyping solutions. In terms of detection, we have been offering a reliable, accurate, and high-quality COVID-19 testing through RT-PCR, helping you to assess emerging mutations' impact on performance claims. Again, we want to be your single trusted source for service and support by ensuring you a secure supply chain that meet your testing demand. Lastly, I would like to also introduce to you the Techman SARS-CoV-2 mutation panel, which offers convenient, customizable real-time PCR research solution for detection, analysis, and surveillance of the SARS-CoV-2 variant. You may choose from a menu of verified real-time PCR assays to build your own custom SARS-CoV-2 mutation panel and get the results you need today while preparing you for what is to come. These scalable solutions lets you to run a few or hundreds of samples to identify one or many mutations, all on your current real-time PCR instrumentation. The workflow for our mutation panel is fairly straightforward. As you can see, following your regular PCR detection workflow, you may move to the confirmation protocol by using the same RNA extract to confirm mutations associated with the SARS-CoV-2 emerging variants. We have quickly launched a few made-to-order assays also to help streamline single nucleotide polymorphism genotyping of the Omicron as well as the Delta Plus variant. I hope this session gave you an overview for today's webinar. We can't wait to share more details with you in the next 2 sessions. You may visit thermofisher.com/COVID-19 mutations to learn more. With that, I thank you for your attention.
Vâng, xin cảm ơn ông Christopher về phần trình bày vừa rồi. Nếu quý vị có bất cứ câu hỏi nào về cái bài thuyết trình vừa rồi, xin mời quý vị nhắn câu hỏi vào mục chat hoặc tham gia tương tác với ông Chris trong phần thảo luận ở cuối chương trình. Các câu hỏi trong mục chat chúng tôi xin phép sẽ giải đáp trong phần thảo luận. Bài báo cáo tiếp theo có nội dung về giải pháp điện di mao quản hay giải trình tự gen Sanger để giám sát, định chủng và phát hiện biến thể Omicron. The next talk will be Sanger sequencing for SARS-CoV-2 surveillance, verification of variants, and detection.
Hello, my name is Steve Jackson, and I'll be your host for the 4th and final module in the SARS-CoV-2 surveillance. Protocol descriptions. In this final module, we will be discussing the protocol that we designed that will allow customers to sequence any position in the SARS-CoV-2 genome by Sanger sequencing. We'll present the browsers that we've used for finding primers, and also a brief description about the data analysis tools that we've presented for customers as well. Okay, so to summarize, what we've done in these 4 modules is provide 4 different protocols for SARS-CoV-2 surveillance. In the first 3 protocols, or in in the first 3 modules, and I think it was in the 3rd one, we discussed the 3 protocols that we previously released for sequencing the S gene dropouts, for focusing on B117 and B1351 strains, and for sequencing the entire S gene and the S gene alone. This 4th protocol that I described for you is the most flexible, and is geared towards letting customers sequence any place they need to in the SARS-CoV-2 genome. We described for them tools that allow them to pick up the primers that they need for sequencing, for running the chemistry, and for if they have Variant Reporter or SISC for doing some variant analysis and identifying if any of the mutations that they identify in their experiment map to the mutations that have been identified in variants of concern. And so we hope this gives customers the most flexibility to identify strains present, either existing strains or new ones that might arise, and help control the progress of the pandemic. So thank you very much. This protocol is meant to complement the other ones that we have posted on our website at the beginning of the year. In particular, this protocol gives customers the flexibility to choose primer sequences that will sequence any region of the virus, not just those found in common variants or the S gene. This is because new variants will arise all the time, and customers need to be flexible in order to sequence the regions where these new mutations arise. And so this protocol provides two tools for choosing wet lab-tested Sanger sequencing primers that can be used to sequence any place, any location, any coordinate in the SARS-CoV-2 genome. Included in these tools are primer sets that are listing the variants of concern. These are preloaded into the tools, and customers can then quickly download the primers that are associated with the designated variants of concern or variants of consequence. And if we check the list of variants of concern in the tools every 2 weeks and update them as needed. The protocol also provides details for sequencing with big dye direct and big dye exterminator chemistries, as the other protocols did. And we also provide in the protocol starting information and basic files that can be used for variant analysis using Variant Reporter or SISC software. The protocols and tools can be found here. The website for the Sanger sequencing page is at the top. And that Sanger sequencing SARS-CoV-2 research page then has links to all the other protocols in addition to this one. Within that page is the links to the visual browser and the lookup table. Both of those then can be accessed from that Sanger sequencing page. So the basis of this protocol, or the basis of this tool, is based on two web-based primer lookup tools that we designed. One of them is a visual browser, and the other one is basically a lookup table. We'll have more to say about that in a second. In addition, though, this protocol also includes a detailed protocol that's written to use the lookup tools to find primers necessary, and also use the big dye direct and big dye exterminator sequencing kits. We have some quick reference guides that allow our customers to perform variant analysis using the results they get from the protocol. And finally, on our web page, the Sanger Sequencing Solutions for SARS-CoV-2 Research, we will have links for the protocol, for the browsers, and for the variant analysis protocols, and all the associated files necessary for that as well. Everything should be on this one page. So to start off with, we'll talk about the visual browser. This viewer will allow the customer to look at the entire SARS-CoV-2 genome. The viewer was built using the JBrowse backbone and should be able to work in any browser. The visual browser is basically broken up into three different boxes highlighted with yellow. On the leftmost side is a list of the available tracks that could be loaded into the viewer. On the top long rectangle is a navigation tool that allows you to scroll back and forth across the genome, zoom in and out. It shows you which reference sequence has been highlighted. It allows you to enter coordinates based on the NC045512.2 genome standard, and also allows you to use a highlighting tool if you need to highlight particular regions of the genome. Finally, on the bottom large square is the actual feature viewing area. By the way, this particular browser will be preloaded with the Wuhan original strain, the NC045512.2 reference genome. So to see features in the browser, you basically choose which tracks you want to visualize. In this first example here, we have the genes and coordinates box checked, and therefore in the viewing area on the browser, you can see that we have the different genes that are in the SARS-CoV-2 genome annotated. You can see they're kind of like those bars with the names of the genes below them. In the bottom browser now, we have clicked on the B117 variant track, In this case, this track has been preloaded with all of the different mutations that have been defined as part of the B117 strain. The browser will become preloaded with variants that are of concern to the CDC and outbreak.org. I'm sorry, outbreak.info. And therefore, the customers can visualize the locations of mutations in variants of concern. By clicking on the different pools, the CDC pool 1 through 6, as shown here on this slide, the positions of the different Sanger sequencing primers can be seen. Here we have a region between about 13,000 and about 16,000 highlighted. And here you can see the positions of the different primers that have been designed to Sanger sequence these particular regions. You can see there's both a left and a right primer that can be used to Sanger sequence across that particular region. Most of these primers are built to be in pairs. They can sequence between 500 and 600 nucleotides about. Although in some cases, you will see, you can maybe customers can maybe choose non-numeric pairs to sequence smaller regions if they want to. In any event, choosing the pool tracks will place the primer positions in the viewer sequence. By the way, the pools that are present here in this example are not the ones going to be in the final viewer. That is upcoming. So the actual tracks and the pools will look a little bit different from this, but basically the way it works will be the same. I also want to emphasize that all the primers that appear in this browser will have been wet lab tested, and only the primers that have worked will be loaded into the browser for our customers. We have found that some of the primers that were designated by the CDC did not perform very well in the Sanger sequencing protocol that we designed. So we designed some new ones, and those will be the ones that are loaded into the browser when appropriate. On the bottom now, we can see that the view, if we zoom in quite a bit, I think this is the most zoomed in view here. You can see the close-up of the sequence with the colored bars, the colored nucleotides. You can see the amino acid sequences in all 6 reading frames. The browser in the final form will have only one of the either forward or reverse reading frame shown, so the customers can see the translation. In addition, you can see the 2 primers, the SC2M139 left and SC2M138 right, are also visible here. And as you can tell, these are from 2 different pairs, the 39 and 38. The arrows will point to the direction of the primers, 5' to 3' orientation. And so you can use the 39 and 38, 39 left and 38 right primers to Sanger sequence, but it'll produce only about 120 nucleotides of sequence there, and might not cover the region that the customer's interested in. So it's better to pick the Sanger sequencing primers that are a numeric matched pair. So to find primers that will cover a particular mutation, what we've done is you can highlight multiple tracks here. So what we've done is we've highlighted the B117 track and the CDC pool primer tracks along with the gene coordinates. The genes are indicated on the top of the file here with the bars. You can see the C14676T synonymous mutation in the B117 mutation, or in B117 strain in this view. And you can also see that the 2 primers, SC2M138 left and SC2M138 right, will sequence across the region that covers the C14676T mutation. And so by choosing those 2 primer pairs, the customer will be able to then design or use these primers to sequence and detect the mutation in the B117 strain. By doing this across all the different mutations or the different coordinates the customers want to use, then they can find the primers that are of interest to them. To get the actual primer sequence, then, what they have to do is they have to click on the feature. Any of the features that are here can be clicked, and a window like you see here will be shown. with the name, the position, the length of the sequence that's there, and the in the box down at the bottom, you'll have the FASTA file showing the sequence itself. Here, the SC2M138 left sequence is highlighted in the light blue. It's only 25 nucleotides long, so it's no problem getting that into one window. And then to order this primer, then the customer would have to copy that primer sequence from the FASTA file, or download the FASTA file, and blast it or put it into an oligo ordering program or something like that. I should also note that this visual browser, although it has the primer sequences, they are not M13 tagged, and if customers want to order M13 tagged primers, they'll have to paste the M13 sequences in manually. The other browser that we have is a text-based lookup app. Like the visual browser, this lookup app can be used with any browser, and it allows our customers then to enter the information in the boxes shown here in order to find the primers that are appropriate for their experiment. So one of the ways that or a couple of the ways that the lookup browser can work is by entering particular strain names or gene names, amino acid changes, genomic coordinates, primer pairs, and they can add the M13 tags to their choices, as you'll see in a second. What I have highlighted here are the strains, the B1351, 117, etc. These strains, the mutations in these strains, are preloaded into the browser, and if the customer wants to order all the primers necessary to sequence B117, they can do that very quickly here. The other option is by gene, and so, for example, if customers want to order all the primers that would allow them to sequence the N gene, they can choose the N, make the gene N choice in this screen here. I also want to note here that this is you can only see one of these two drop-down menus at a time. The customers will not be able to visualize both menus as shown in this slide. This is just, for example, illustrative purposes only. As I mentioned, this lookup table will allow customers to type in the genomic coordinates. Here, the customer has typed in 24567 as the location in the reference genome that they want to query. And then when they hit the search tag and clicking the M13 box, then they will then get a list of the primers that can cover that particular coordinate. Here, there are two sets of primers, SC2M162 left and right, and SC2M163 left and right, that will allow the customer to sequence this particular region. Either of these should be able to work. When customers want to look up particular amino acid changes, like for example the E44K, which is commonly seen in some of these highly transmissible variants, they will need to put in the gene name and the amino acid change. For example, here we know the E484K is in the S gene, and so they choose the S gene 484K. And then when they click on search, then the primers that will cover that particular information will be found. Again, here we have the SC2M158 left and right, which allow the customers then to sequence across the coordinates that encompass the E484K mutation. So when customers are ready to export their primer choices, they can click on the export to order button. And what will happen is that the browser will then generate a text file loadable in Excel, which will have all the primer sequences, the names, the synthesis scale, and the purification choices already made. All the customer will have to do then is to type in their name, in this case Jane Doe, and then the text file is then uploadable into our oligo ordering page. We have links to that on the Sanger sequencing webpage, as I alluded to before. Note here the synthesis scale is preloaded by default at 25 nanomolar, and the purification is desalted. If the customer wants to change that, they can, although they would have to check to make sure that the scales and the purification choices are correct for our oligo ordering service. Nevertheless, the 25 nanomolar scale and desalted is entirely appropriate for sequencing reactions in this protocol. And the 25 nanomolar scale will produce a large amount of primers and can be used quite a bit for several hundreds to maybe 1,000 different sequencing reactions. Okay, in addition to the protocol and the browsers, the fourth module includes a guide to variant analysis using 2 of our software packages. One of these is Variant Reporter, and so we have written a brief user guide that allows customers then to take their sequencing results and see whether or not any of the variants that have been defined in the variants of concern are present. So we provide a brief user guide. It's a brief user guide for customers to follow. We have a text file that contains the mutations that are commonly seen in variants of concern that can be uploaded into Variant Reporter. We have a text file that is of the primer sequences that are in the protocol. And finally, there's a FASTA file for the virus reference sequences. All of these will be downloadable on our web page, and customers can then load these into Variant Reporter according to the directions that we have in the protocol. I want to also point out that the user guide that we supply is just a getting started guide. It is not meant to be comprehensive or a complete description of all the features available in our software. Customers who want to have a complete description of all the features should look at the original documentation that came with the software when they purchased it. One of the other important things that is often done with variants is that the sequences that are made are tracked by various global entities like GSAID or the Genome Detective. And so we also provide in the guidelines instructions for exporting the results from these experiments so they can be uploaded then into external databases for tracking and surveillance globally. Again, we don't have direct links into these different sites, but we do provide a guidance for customers to export the data that they generate so that they can be uploaded to these external sites for tracking purposes. So I just described Variant Reporter. We have very similar sets of things for SISC for customers that have SISC software. We have the quick reference guide, as I described for them, that allows them to get started. We have supporting files that will be provided that allow, it's a demo project that has the reference sequence, primers, and the variants of concern mutations preloaded into this demo file so that customers basically just have to download the demo file, and they can then upload or interpret their own sequencing results in the SISC environment. And like I mentioned, we also have some guidelines for exporting data so it can be uploaded into external databases. The last thing I want to mention is throughput. We often get asked about what the throughput for these different protocols are with the PCR, the Big Die Direct, the Big Die Exterminator, and CE. And so I've put together some guidelines here for throughput on our different platforms, on SISC Studio, on 3500, on 3500 XL, and on the 3730 XL. With the exception of SISC Studio, all these have a 50 centimeter capillary installed. So to amplify the sequencing amplicons, it takes about 1 hour to run that, no matter what the instrument is. And then for the cycle sequencing and Big Die Exterminator, it takes another 2 hours. So a total of about 3 hours to do the PCR and cycle sequencing and cleanup. And then running on the different CE instruments takes various amounts of time. In general, for running a single set of reactions, a single injection, takes about 40 minutes on any of our platforms. And so, for example, on a SISC Studio, I see that per sample it takes about 3 hours and a minute to run, and that's not quite right. It should be 3 hours and 41 minutes to run a single sample on SISC Studio. On SISC Studio, to run an entire 96 well plate, it takes about 20 hours or so. On a 3500 for single sample, it takes about 3 hours and 44 minutes, et cetera. And so you can go through this table and find out, how long it takes to run either a single sample or a single injection, or a 96 well plate that's completely loaded up with samples. And then based on that, we calculated, how many samples a user might be able to run in a shift, on an 8-hour shift, for that is. And so if you keep the SISC Studio going, the customer, a customer can run about 14 samples in a 96 well plate in an 8-hour shift. That includes the Amplicon PCR, the Dig-Dye Exterminator cycle sequencing, and the CE running time. On a 3500 with an 8 capillary, it can run about 27 samples in an hour, or I'm sorry, in a shift. On a 3500 XL, about 81 samples in a shift. And on a 3730 XL, that is 96 well, 96 capillary array installed there, the customer can run about 320 samples in an 8-hour shift if they keep the machine fed. In other words, they would have to set up multiple 96 well plates over the course of that 8-hour shift in order to achieve this throughput. So, and these are just guidelines. Some of the actual performance might vary a little bit.
Vâng, đó là các hướng dẫn rất là kỹ càng về việc thao tác cần thiết để thiết kế mồi và phân tích kết quả giải trình tự Sanger phục vụ cho nghiên cứu virus SARS-CoV-2. Xin cảm ơn hãng Thermo Fisher. Nếu quý vị có bất cứ câu hỏi gì, xin đừng ngần ngại nhập vào phần chat để chúng tôi có thể giải đáp trong một thảo luận tiếp theo. Ở bài thuyết trình cuối cùng chúng tôi muốn giới thiệu đến quý vị hôm nay đó là về giải pháp giải trình tự gen thế hệ mới NGS trong việc nghiên cứu và phát hiện các biến chủng SARS-CoV-2, trình bày bởi cô Tan Hoi Tran, giám đốc sản phẩm của hãng Thermo Fisher. The next and the last presentation of today is about next generation sequencing, the ion anti-6 SARS-CoV-2 inside research assays for Omicron variants.
Thank you for listening to the presentations of fast confirmations of Omicron variants with the ion amplific SARS-CoV-2 Inside Research Assay brought to you by Thermo Fisher Scientific. Before we begin, a quick introduction about myself. My name is GC. I'm the product manager representing Thermo Fisher Scientific. Prior to joining Thermo Fisher Scientific, I have more than 7 years of experience in the provisions of scientific applications, in-depth technical support, and customer training for real-time PCR, capillary electrophoresis, microarray, and next-generation sequencing covering the Asia-Pacific region. First of all, I would like to provide a fast facts on the Omicron variants. The Omicron variants, or B11529, is a SARS-CoV-2 variant. It was first identified in the South Africa. It has been designated as variants of concern by World Health Organizations. Omicron has an unusually large number of mutations and appears to be highly transmissible based on some early reports. The emergence of the Omicron variants has already led to a wave of lockdowns and travel restrictions to mitigate the alarming spread of the variants. One of our Thermo Fisher Scientific customer, who is Fisher here, her name is Dr. Alison Glass. Dr. Alison works in Lanchurch Laboratories in South Africa. She is running the Amplific SARS-CoV-2 solutions with the Genexus integrated sequencer, and she is one of the first to discover the fact that there might be a new variant circulating in the community. Dr. Allison was collecting PCR samples from mid-November, and she observed S-gene dropout in PCR results using our Applied Biosystem SARS-CoV-2 PCR kits, suggesting a possible variant. By sequencing these samples collected from November 12 to 20 in-house with the Ion Torrent Genexus integrated sequencer, Dr. Ellison and her team were able to analyze and identify the specific virus mutations in less than 24 hours and alert the South African National Surveillance Program for confirmations and immediate actions. The B11529 reported to WHO on November 24, and on November 26, B11529 is designated as variants of concerns and named Omicrons. In summary, it took about a week from the detections of B11529 sequence to the declarations of an Omicron variant, which is a record time much faster than we have seen for any of the reports of other variants. Part of the reason is due to the fact that she has the GeneXus instrument available in her lab to enable her to rapidly sequence those samples and get results within 24 hours. The variants of concern Omicrons belongs to the Pangol lineage B11529, characterized by 45 to 52 amino acid changes, including 26 to 32 in the spike proteins compared to the reference strains. The Omicron has a long list of mutations, some 50 in all. Of these, 32 pertain to the virus spike proteins, a vital part that the virus uses to gain entry into the host cells and which is also the target of most COVID-19 vaccines. Part of the reason why it was concerning is because the number of mutations in the spike protein. Omicron has 32 mutations versus 13 mutations in Delta. There's a lot of scrutiny around the spike protein because it is essential for the virus ability to infect cells, so lots of disease-fighting antibodies can neutralize the virus targeting those spike proteins. If there's a mutation that changes the amino acid in a way that can alter the structure or chemistry of the proteins, it can prevent the antibody from binding as they normally would. However, no one knows how multiple mutations work together, and researchers are still studying it. The implicit SARS-CoV-2 Insight Research Assay is an enhanced assay to survey the complete SARS-CoV-2 genome for epidemiological investigations. The assay consists a total number of 242 unique amplicons across two pools, with 237 amplicons specific to SARS-CoV-2, with the highlight of the additions of variance tolerance primer for emerging variants, as well as 5 newly designed human expression control targets for sample confirmations and quality assessment. The assay covers over 99% of the SARS-CoV-2 genomes. The assay amplicons range from 125 to 275 base pair with significant overlap. It is this overlap that gives exceptional performance over time against naturally occurring variations. In the case of the Omicron variants, I'm happy to share that our ion implicit SARS-CoV-2 Insight Research Assay can enable the confident identifications of the Omicron variants due to the robust coverage provided by the variance tolerance primers. Here's an example to showcase the capability of the implicit SARS-CoV-2 Insight Research Assay in obtaining the complete sequences for emerging variants with the intelligent panel design. In this figure, the upper parts refer to the coverage of the Twist Biosciences SARS-CoV-2 Control 1 amplified with our original SARS-CoV-2 variants, SARS-CoV-2 or community panels. And the bottom part refers to the coverage of the Twist Bioscience SARS-CoV-2 Control 1 amplified with our latest implicit Insight Research Assay panel. With these twist control viral variants that has a mutation in the original panel primer region, you can see that we managed to rescue the library amplicons with the Insight panel due to the additions of the variance tolerance primers, resulting in the improved coverage. In this study, we are looking into the performance of the implicit SARS-CoV-2 assay relative to competitor assays. Result shows that ion torrent has a higher percentage identified bases in submitted sequence, indicating higher tolerance for new mutations due to the intelligent panel design, which reduced the number of amplicons dropouts. When a base cannot be called based on the sequencing data, instead of A, T, C, or G, and N is inserted. We can quantify how well an assay is performing by comparing the percent of the identified or non-N bases. For US submitted ion torrent genomes in GSET, we see an average of 99.85% of bases identified, compared to 98.83% in competitor assays. As demonstrated in the box plot on the right under the figure 6, we also see much fewer low quality or high-end samples submitted using the ion torrent technology compared to our competitor, which have a much wider range of the percentage non-N. Despite the manifold increase in the number of available genomes, the panel continues to successfully amplify and assemble whole genomes. This study here is based on the data collected from the Omicron sequences submitted in GSET back in 2nd December 2021. The raw data are available in SRA, which is another public database of the SARS-CoV-2 sequences, where key mutations were not identified in 61 out of 279 Omicron samples because of the dropouts caused by the Arctic version 4 protocols and primer set. As you can see from the table here, although ion torrent technology submitted less sequences back in December compared to competitors' technology, we have a very low percentage N for no-call, which is 0.35% as compared to competitors which reported 1.12% and 2.25% respectively of the average N percentage. With a high degree of functional redundancy, the panel has continued to prove that it is robust to the natural variations, allowing for overall sequencing quality higher than competitor assays. In addition, the performance of the assay was confirmed by multiple sites as the intelligent design of the assay provides coverage to identify Omicrons as well as previous VOCs and VOIs. In extent to the previous slides, in this graph, we have tabulated how many times each sequencing technology was able to call either wild type, mutant, or no-call for the spike mutants. Again, ion torrent technology provides best coverage of spike proteins with average 1.85% of no-call permutations in the spike proteins as compared to 8.58% and 6.61% respectively in competitors' platform. This information is particularly important as the Omicron has a long list of mutations, some 50 in all. Of these, 32 pertains to the virus spike proteins. Here we showcase how ion torrent technology leads the sequencing performance with the lowest percentage of no-call in spike proteins. With our implicit SARS-CoV-2 Insight Research Assay, we provide the comprehensive informatic suite for seamless informatics and data uploading to the public SARS-CoV-2 data repositories. After the sequencing run is completed, the analysis through this plugin are automatically initiated, providing a complete workflow from designs to interpretations, analysis, and data upload with minimal hands-on time. The plugin includes the SARS-CoV-2 coverage analysis and variant caller, which provides statistics and interactive visualizations of coverage over targeted regions of the SARS-CoV-2 reference genome, as well as SNP and indel calling at a given positions. For various annotations, we have the SARS-CoV-2 annotate SNP-F. These plugins text the variants detected by the variant caller and use the SNP-F to produce a list of annotated variants and predict the effects of the genetic variants on genes and proteins with reference to the public or the private database. We also include the plugin to generate consensus sequence. The generate consensus plugin align reads to the reference, identify low frequency variants, and eventually provided consensus sequence in FASTA format. And you can then opt to upload the FASTA sequence to public repositories for global surveillance. In addition, we have integrated the lineage assignment plugin. This plugin assigns a SARS-CoV-2 Pango lineage to its consensus FASTA sequences output by the Generate Consensus plugin and the Pangolin program. Monthly updates on this plugin is expected as well to keep the database up to date. Here's another customer success story featuring Dr. Eva. She sequenced the first Omicron case in Czech Republic. We have a great quote from her talking about the use of the implicit SARS-CoV-2 insight research assay together with the ion torrent genexus integrated sequences, which enable her to confirm the new Omicron strain in less than 24 hours. We do have a blog post here to provide more insights into customer success story on the use of implicit SARS-CoV-2 insight assay to sequence and identify the SARS-CoV-2 variants. For example, the work of Dr. Ellison, which we mentioned earlier, who is one of the first to detect these new variants at Lancet Laboratories in South Africa, and Dr. Eva's work in the detect of the first Omicron case in the Czech Republic. There are more ion implicit SARS-CoV-2 resources available in our Thermo Fisher website. You can go to the browser and type in thermofisher.com/SARS-CoV-2 insight. In this webpage, you can access to more information and NGS educational resources for SARS-CoV-2 research, the ordering informations, recent customer talks and webinar, as well as our customer experience and success story. If you do have any questions, please reach out to us and we are happy to hear from you. And we have also included the links to the additional resources in this slide, including the documents released by the WHO and ECDC on the classifications of the Omicrons, the implications of emergence and spread of the Omicron for the EU or EEA, as well as the Africa Centers for Disease Control and Prevention statement regarding the Omicrons. There are also links to the press release, blog posts, and user guides in regards to the topics of applied biosystem taqpath COVID-19 test and the ion implicit SARS-CoV-2 insight research essay. With that, I thank you for your attention for listening to these presentations. Have a nice day.
Vâng, thưa quý vị, đó là tất cả các nội dung báo cáo trong buổi hội thảo ngày hôm nay của chúng tôi. Nếu quý vị quan tâm và muốn nhận các tài liệu liên quan đến buổi thuyết trình này, xin mời đăng ký nhận thông tin thông qua đường link trong chat. Ở mục tiếp theo, trình hội thảo, chúng tôi xin trân trọng kính mời tiến sĩ Phạm Thu Hằng, Giám đốc Trung tâm CGG Center sẽ dẫn dắt phần hội thảo ngày hôm nay cùng với đại diện hãng Thermo Fisher là ông. Đó là tất cả các nội dung báo cáo trong buổi hôm nay của chúng tôi, thưa quý vị. Nếu quý vị quan tâm và muốn nhận các tài liệu liên quan đến buổi thuyết trình, xin mời đăng ký nhận thông tin thông qua đường link chat box. Tiếp theo chương trình hội thảo là phần thảo luận sẽ được dẫn dắt bởi tiến sĩ Phạm Thu Hằng, Giám đốc Trung tâm CGG Center. Xin mời tiến sĩ Phạm Thu Hằng. Dạ, xin mời tiến sĩ Phạm Thu Hằng bấm mic ạ.
Xin chào các anh, các chị và các bạn. Trước tiên là tôi xin cảm ơn công ty SISC đã tổ chức buổi hội thảo ngày hôm nay với một chủ đề rất là thú vị và cũng được sự quan tâm của rất là nhiều người, đặc biệt là liên quan đến biến chủng Omicron. Hiện nay đây là cái mối quan tâm lớn của không chỉ ở Việt Nam mà trên thế giới. Thì cái câu hỏi thường trực đặt ra với những người mà làm xét nghiệm cũng như là nghiên cứu và liên quan đến các kỹ thuật sinh học phân tử của chúng tôi hiện nay là làm thế nào để mà có thể xác định được cái biến chủng này và bởi vì là cái biến chủng này lây lan rất nhanh, nên là các cái kỹ thuật mà được áp dụng ngay lập tức thì sẽ là những cái công cụ hiệu quả để mà hỗ trợ chúng ta trong việc phát hiện và phòng chống dịch hiện nay. Thì qua bài nói chuyện của Thermo Fisher thì tôi thấy hãng đã đưa ra những cái giải pháp chính liên quan đến sinh học phân tử, từ những cái giải pháp sử dụng công nghệ cao như là NGS để giải trình tự, hay là Sanger sequencing nhằm giải trình tự, và đến những cái kỹ thuật mà hiện nay là đang phổ biến được sử dụng nhất, đó là kỹ thuật realtime RT-PCR. Thì ở ngay đầu buổi nói chuyện bên SISC cũng đã nói đến cái việc là sáng nay thì đã công bố 3 ca Omicron trong cộng đồng, và bệnh viện 30 tháng 4 cũng nhờ sử dụng kit taqpath của Thermo mà phát hiện cái việc gen S có những tín hiệu của dropout, và từ đấy giải trình tự phát hiện ra cái 3 trường hợp này. Thì đây là một cái tin rất là đáng mừng. Vì vậy nếu mà các anh các chị có câu hỏi gì có thể đưa lên phần chat, hoặc là các anh chị có thể tương tác trực tiếp với ông Christopher đây, và tôi sẽ hỗ trợ dịch tiếng Anh và tiếng Việt ạ. Hi Chris.
Hello, good afternoon.
Yeah, hi.
Hi, Dr. Han.
First of all, thank you very much for your talks today. It was very interesting for me, and I think also for all the other audience. So I see that there are a few questions on the meeting chat now.
Okay.
Can I just translate for you, and can you please answer the question, please?
Yes, sure. Thank you.
So the first question is, what is the first negative when using Thermo kit to detect Omicron?
What is the false negative?
Yes, false negative. Great.
Okay. Okay. So our taqpath COVID-19 test, which is a Thermo Fisher test kit, has been tested by a lot of other clinicians and also researchers around the world. Okay. So we actually obtain a very low to no false negative results from our test kit. And we also released another technical bulletin when we compare Thermo Fisher kit with Roche. Okay, so in this study, the Roche kit actually show 13 false negative results out of 400 over samples that we actually tested. So our kit actually show a very low percentage of false negative in our test kit. I can share with you the link also to the paper.
Yes. Do you want to show on the screen?
Yeah, sure. Just give me a moment.
Trong lúc anh Chris chuẩn bị để chiếu lên màn hình cái bài báo nghiên cứu của hãng về việc sử dụng kit này, thì tôi tạm dịch qua câu trả lời của anh Chris vừa rồi. Như là anh có nói đến việc là họ đã làm cái thử nghiệm ở trên hơn 440 mẫu, thì và họ cũng so sánh với cả cái kit taqpath của hãng Roche, thì họ thấy là cái tỷ lệ âm tính giả của kit taqpath này là rất thấp, và gần như là không phát hiện âm tính giả, từ thấp đến không có âm tính giả. Và anh sẽ show cái bài báo này lên trên màn hình ạ.
Alo anh Đạt, em Dung anh Đạt. Anh dạ anh cho con xin một cái tờ giấy để đặt cho con nhỏ bé nó ở.
Just give me a moment, yeah. Okay, I'm opening it right now.
Trong thời gian chờ đợi các anh chị nào có câu hỏi muốn hỏi chuyên gia thì các anh chị có thể đưa lên mục chat để chúng tôi có thể tương tác và hỗ trợ trả lời các anh chị ạ.
Sorry, I think my file is loading. Just give me a moment more. Maybe we can proceed to the next question first before I show and answer the first question. Dr. Hang?
Yes.
Yeah, while waiting for the page to load.
So
yeah.
The next question is, can the real-time RT-PCR detect the Omicron variant or not? Please.
Okay, so the taqpath real-time PCR will actually give you an early identification about Omicron variant. So for you to confirm the variant, we also have the mutation assay that helps you to confirm the Omicron variant. Yeah, so by using the taqpath kit, you will first see the S gene dropout. So again, our test kit, we have 3 targets, right? We have the operating frame 1AB, we have the S gene, and we have the N gene. So when you detect only the N gene and the operating frame 1N gene without the S gene, then you will roughly know that it could be an Omicron sample.
Yes. Chúng ta có thể sử dụng real-time PCR, sử dụng kit taqpath để xác định được để phát hiện cái biến chủng Omicron. Nhờ vào việc là kit taqpath này thì có 3 gen là gen S, gen N và gen ORS. Thì khi mà đối với những mẫu mà thấy tín hiệu của gen S không xuất hiện mà chỉ thấy tín hiệu của gen N và gen ORS thì chúng ta có thể nghĩ đến việc đây là biến chủng Omicron. Và để mà xác định được chính xác đây là biến chủng này thì theo tôi hiểu là phải làm giải trình tự sau đó. Tuy nhiên là bằng cái phương pháp sử dụng cái kit taqpath này thì chúng ta đã có được cái dấu hiệu đấy là Omicron và nghi ngờ đấy là Omicron để có thể tiếp tục xác định xem chính xác nó hay không. Và ngoài ra thì taqman còn có những có 3 kit khác. Chris, you mentioned that taqman have 3 different Omicron specific markers, right?
Yes, we have. Yes.
Could you specify it? Is that a kit or a combination of probe and primers that we can order?
Yes, sure. I can show you now. Okay, before I show you the mutation, let me back to the first question first. All right. Okay, let me show you the first question. Okay, I hope you can see my slide now. Can you?
Yes.
Okay. Okay, so we actually have a lot of comparative evaluation that show you that shows you the superior clinical performance of our assay. So this paper, published in Singapore, that shows you taqpath with 100% of the sensitivity when you compare it with other vendors like Fortitude, Realstar, Cobas, which has less than 100%. So for this one, there is also another evaluation that exhibit excellent performance with positive and negative percent agreement of 100% for the taqpath combo kit when you compare it to the Surfit Gene Expert assay. So this is when we compare the taqpath with the Gene Expert. All right. And another one, so this one is also for pooling. I understand that the Vietnam market, regularly do pooling for this one. So this one, we also demonstrated 100% sensitivity and specificity, and with the limit of detection of 2 copies per microliter. Okay, so this is all published by an external researcher. I can share this with you also if you want. And we are also being used as a standard of care PCR by the UK government, as you can see here. So this is we are the standard. We are the gold standard for UK government for in terms of PCR testing. And last but not least, this is what I mentioned just now. We demonstrate we demonstrated a positive predictive agreement of 100% and also the NPA of 100% also when we compare to Roche-Cobas. So we tested a total of 446 samples and we found 13 false negative results for Roche-Cobas test. So this show you how great is the performance of our taqpath COVID-19 kit. Okay, so I hope I did answer you questions about false negative results. I can share all this paper with you after this webinar, no problem. Okay. Yeah.
Yes, I try to translate those
okay.
4-5 slides.
Okay.
Okay. So trên màn hình này thì đây là một cái nghiên cứu của hãng và sử dụng cái kit real-time PCR của các hãng khác nhau. Như các anh các chị có thể nhìn thấy ở đây, họ sử dụng hãng của Fortitude, Greenstar, Cobas hay là taqpath, thì trong đó taqpath của hãng Thermo đang là có tỷ lệ phát hiện cao nhất là 100% và Cobas là 95%. Next, please. Next slide, please. Please.
Yeah, I'm on the next slide already. Are you seeing the gene expert one, or is still the same slide?
The next slide. The next paper, please.
What about now?
Yes. Okay. Và họ có một nghiên cứu ở đây sử dụng cái kit taqpath này thì cũng cho thấy là có độ nhạy là 100%, độ nhạy, độ đặc hiệu, độ chính xác là 100% trong cái bài báo đăng trên tạp chí này. Và ngưỡng phát hiện là 2 copy trên một microliter. Đây là một cái ngưỡng khá là thấp theo như tôi biết so với các kit khác. Next, please. Okay. Can you repeat this?
Okay, so the UK government has been using the taqpath test when they compare with other brands. So they use our taqpath test as the standard of care and as the gold standard PCR.
Okay. So chính phủ của chính phủ Anh thì đã sử dụng taqpath để so sánh với các kit khác và họ đã sử dụng cái kit taqpath như là một cái kit chuẩn trong cái xét nghiệm PCR hiện nay.
Yeah, so that means they use the taqpath test as the reference test. Yeah. Next slide, the last slide.
Last slide. Yeah. Đây là một cái nghiên cứu khác đã được công bố trên tạp chí này, thì họ đã làm trên 446 mẫu và họ thấy rằng với kit của Roche thì có 13 mẫu là bị âm tính giả. Trong đấy thì với kit taqpath của Thermo thì đạt tỷ lệ là phát hiện được 100%. Yeah.
Yep.
Yes, thank you.
Okay.
So, relate to my question before, could you please specify the Omicron specific markers that Thermo Fisher has?
Yes, sure.
Yep.
Okay, let me show you another slide. Okay, so can you see my slide, my web page here, the screen here?
Yes.
Okay, so this is what I mentioned just now. Okay, so we have a detectment mutation panel to help you to differentiate between all these variants. Okay, so if you scroll down, we have more than 50 assays, 50 real-time PCR assays to help you to target whichever variant that you want to study. Okay, so for example, okay, you see this is a new Omicron variant mutation assay. So we have 5 here. Okay, the G339D, this one, the second, third, as these are the 5 Omicron related marker for Omicron assay. Okay, and this paper, when you click on this paper, okay, so this paper actually set up a 4 marker panel to help them to differentiate between the Omicron variant as well as the Delta variant. Okay, maybe Dr. Hang, you want to translate first, then I continue.
Ok, như các anh chị có thể thấy trên màn hình ạ, đây là các cái đột biến, là 5 cái đột biến của biến chủng Omicron, thì họ có chỉ 5 cái đột biến ở trên màn hình này, thì hãng Thermo sử dụng 4 trong 5 đột biến này để phát triển cái bộ marker của họ. Yes.
Okay, yeah. So for this one here, if you scroll down further, you can choose. Okay, so this will be your mutation, and what is the variant, and what is the lineage. Okay, and when is it being identified? Which country first? So all of this here, you can see the type of mutation here and what variant that are responsible for this mutation. Okay, so in this website, you can find a lot of information. So you see here, this is the Omicron, okay, and we have more actually. Here also, this one is also for the Omicron variant, okay, and we have the Omicron variant, and this is for the Delta variant. All right.
Nếu anh chị vào trong website này và các anh chị nhìn xuống phía dưới thì kéo xuống phía dưới thì chúng ta có thể thấy là từng đột biến một đã được liệt kê ở đây, và họ sẽ nói cho chúng ta là nó thuộc biến chủng nào và chúng ta quan tâm đến. Yes, please.
Okay, yeah, I think that's all for this mutation one. Any more questions? Is it clear enough?
Yes, I see few more questions.
Yeah, sure.
So, bạn Lương ở đây hỏi là trong một mẫu có xuất hiện biến chủng Omicron bằng giải trình tự NGS có phát hiện được không? Để phát hiện thì nồng độ tối thiểu của biến chủng Omicron có trong mẫu là bao nhiêu %? Hay nói cách khác là deep read là bao nhiêu % thì xem đó là một đột biến. Okay, this question related to NGS, please.
Okay. Yeah.
So the question is when we do sequencing to detect Omicron by NGS method, what is the minimum concentration of viral RNA needed, or what is the percentage of deep read in order to detect this?
Okay, so we need only as low as 1 nanogram of the RNA sample of the SARS coronavirus 2. And regarding the percentage of the reads, I will need to confirm the details before sharing the details with you. I will ask our channel partners to follow up with you later.
Okay,
yeah, I do not have the answer for that right now. Yes.
Thì anh có thể Chris trả lời là chúng ta chỉ cần một lượng rất là nhỏ, 1 nanogram RNA của virus để thực hiện cái công việc này. Okay. Another question.
Yes.
This. Câu hỏi của bạn Bạch Quỳnh. Vậy tại sao hãng không phát triển bộ kit dành cho việc phát hiện các vùng gen đặc trưng cho biến chủng Omicron thay vì dựa vào sự dropout của gen S? Okay, very interesting questions again, Chris.
Yes.
That why Thermo Fisher does not develop a real-time PCR kit specific to detect Omicron variant, instead of using taqpath kit right now, and just depend on the S gene dropout to detect Omicron. Please.
Okay. So in our research and development, so it really takes time for us to develop an assay and go through the CE-IVD verification. Yep. So it takes time, okay, because the registration is not easy and the extensive validation has to be done also. So I think to respond quickly to the pandemic, I think this is the fastest way that we can have now in order to quickly respond to the Omicron variant detection. Then you use the positive sample for Omicron to do a mutation analysis or sequencing. I think that is the fastest route for now, rather than spending more time to develop a new assay for Omicron only. Yeah.
Câu trả lời của anh là bởi vì là như các bạn đã biết là để phát hiện được cái biến chủng này thì cũng mất thời gian cho nghiên cứu và phát triển để đưa ra được cái bộ kit đặc trưng riêng cho Omicron. Mà hiện nay thì Thermo Fisher đang có cái kit taqpath này là đã có thể dựa vào cái tín hiệu của gen S bị mất đi mà đã có thể nghĩ đến việc đấy là chủng Omicron rồi, và đây là một cái giải pháp mà nhanh nhất mà chúng ta đã có thể sử dụng ngay được. Chris, but my question is,
yes.
So does Thermo have plan to develop the real-time PCR kit which is specific for Omicron variant right now?
Okay, for the detection part, no. Okay, but for the genotyping part, yes. That means the genotyping one is the mutation panel that I showed you earlier. For the real-time PCR for Omicron only, no, we don't have that plan.
Thì câu hỏi của tôi là hãng Thermo có dự định là phát triển kit đặc hiệu để phát hiện biến chủng Omicron hay không, thì anh Chris có trả lời là nếu mà dùng real-time PCR thì họ không có dự định làm việc này, nhưng mà nếu mà dùng thắc nhanh thì họ đã có những cái bộ assay đặc trưng cho nó rồi. So if you want to use it, then we have to order the primer set from you and do the mix ourselves. Is it right?
No. You mean for the taqpath kit?
Mm-hmm.
Yeah, so the taqpath kit actually comes with a kit. You don't have to, it is a CE-IVD kit. So you do not have to do your own master mix, everything. So within that kit, you only have two components. You have the master mix, you have the assay multiplex. When you combine the two with water, then you are good to add your sample and run the PCR. That's simple.
Okay.
Yep.
I see there's another question. So, câu hỏi của anh Duy ở đây ạ là ở Việt Nam thì các bác sĩ làm chẩn đoán thì thường dựa vào cái giá trị CT của gen Y để điều trị các bệnh nhân COVID. Vì cái kit của Thermo Fisher thì không có gen Y, vì vậy mà dựa vào cái giá trị tương đương như thế nào giữa giá trị CT của gen Y và kit taqpath và các cái gen khác trong kit taqpath để mà có thể đánh giá được trong việc điều trị. Okay, so in Vietnam, the clinical doctors use the CT value of E gene for the treatment procedure for COVID-19 patients. But in the Thermo kit, you don't have E gene. So how can we correlate the CT value between E gene from other kit to taqpath, 3 other genes from taqpath, please?
Okay, thanks for the questions. Okay, for quantitative SARS-CoV-2 assay, okay, it doesn't matter whether you use the E gene, you use the N gene, or use the S gene for quantification. Okay, so we have customers at the UK that use the N gene for quantitation. All right, so something that I want to highlight here is that when you use the E gene, for example, if you use the CT value, you need to make sure that you use the same PCR for the same patient to monitor the viral load. Okay, so you do not use E gene at first, then maybe next week you use N gene to check for the viral load. It is not right. You need to use back the same one. So in terms of, there is also a research paper that also indicate that when they compare S gene and N gene for quantitative assay, the performance is about the same. So it doesn't matter whether you use the E gene, the N gene, S gene, or other SARS-CoV-2 genes for quantitation. It is important that you use the same gene for the same patient. And another point I want to highlight is that rather than referring only to the CT value, you need to take note of the housekeeping gene of the patient as well when you do quantitative assay, because you need to correlate with the housekeeping gene of the patient, right? So that will actually show you whether are you correcting the right sample. Okay, then the relative quantification will be more accurate as compared to you looking at the CT value only. Yes.
Yeah, thank you. Very good question. I think this is related to the technique, molecular technique, right?
Yes.
Tôi nghĩ là đây là một câu hỏi rất là thú vị về việc đánh giá và điều trị bệnh. Thì theo như câu trả lời của anh Chris là việc đầu tiên là để đánh giá được nuôi bệnh thì chúng ta phải thứ nhất là dựa vào cái gen housekeeping gene, dựa vào gen nội kiểm, và cái CT của gen đích phải được tính dựa vào cái gen nội kiểm này thì mới đưa được cái độ chính xác nhất. Bởi vì là khi cái lượng mẫu đưa vào nó khác nhau thì cái chỉ số của cái gen nội kiểm cũng như gen đích sẽ bị thay đổi theo. Và điều đặc biệt ở đây là nó không phụ thuộc vào cái gen đích là gen gì, gen không phụ thuộc gen đích là gen N, gen S hay là gen Y, mà quan trọng nhất là chúng ta phải sử dụng một cái gen đích cho chính cái bệnh nhân đấy và sử dụng đúng cái bộ kit đó cho chính cái bệnh nhân đấy trong quá trình theo dõi điều trị thì mới đánh giá được. Đấy là hai cái điểm chính mà anh Chris đưa ra. Và một cái ví dụ mà anh đưa ra đấy là ở nước Anh thì họ không phải là sử dụng gen E mà họ sử dụng gen N để theo dõi bệnh nhân. Thank you, Chris. Các anh các chị có ai muốn tương tác trực tiếp với cả với chuyên gia không ạ?
Hi, Chris.
Hello. Yes.
Hi, Chris. I'm not sure if I missed some information, but you know, for the confirmation test of SARS-CoV-2 virus,
yes,
we may need to run evaluate more than one target, like not only E gene but also RPRD and N or S gene.
Correct.
So in the in the point of view of a physician, right? So we all know that at the CT 30, above 30, the patient does not need to be in hospital anymore. He or she can go home and take care of himself and
for home recovery
regularly to the hospital and so on and so forth. Yep. So my question is that which gene or which CT of what CT of which gene should be considered for that point, which is the point of no longer necessary to be in hospital, right? So I myself here normally consider E gene, right?
Okay.
Even though we have E gene and RPRD.
Yep.
And also when we do experiments or the test, we know that the E gene, the RPRD, the N, and the S gene, they are interact, or they are amplified at a different time point and different CT, yep. Right? So which one is do you think should be considered like the point of CT, which it is important for the for the viral load. Yes.
Okay, so again for this question, thank you for the questions. Again for this one, it depends on the clinicians around different countries. Okay, so what I am seeing in the market right now, or in the world right now, other than e-gene, the doctors refer mostly on the CT value of the N gene actually. Yeah, N gene. So there are some doctors use the RDRP gene also. Some of them use the S gene. Okay, but what they are doing, right, what I see most frequently would be the E or the N gene. Okay, so and by looking at the CT value alone is actually not accurate enough. So you should also, if you really want to quantitate the viral load, right, you need to run also housekeeping gene PCR, for example, the RNA-seq. Okay, the RNA-seq from the same patient. Then you actually apply a formula to do a relative quantification. Why RNA-seq? Because RNA-seq is a housekeeping gene. Okay, then by knowing the level of the CT value of the RNA-seq gene, then we will roughly know the quality of our sample, whether the quality of the sample is good or bad. If you have a very low level of RNA-seq sample, it is still not considered bad, but you can do a relative or comparative CT value study. So a housekeeping gene here is really important if you really want to do a quantitative test.
I see. So can we use another viral RNA which is used for to say measure the efficacy of RNA extraction, like internal control? Some kits they put
MS2.
Yes.
Yes,
yeah,
yes.
Exactly.
Definitely.
So is that possible to use that external RNA or another viral RNA for how to say calibrator or quantification calibrator?
Okay, for if you want to do a quantitation,
yes.
The MS2 may not be the most suitable one. Okay, if you really want to do quantification, then you need to still use a human housekeeping gene for that one.
Oh, I see.
Okay.
Yep.
So the
so that's
external
not applicable
control
for all the tests and all the kits, because yeah, some kits they use human RNA. Yes.
Yeah, because that one is unique to the patient. So we need to see the RNase level of that patient itself. But I have a question for you actually. How do you see the importance of quantitative tests right now for Vietnam? How is it going to be very mainstream in the Vietnam, or just for some cases only?
Actually, from the physician point of view, they are not very much care about the quantitative data.
Right. Yes.
I think the symptom of the patient, the situation of the patient is more important.
Correct.
And the CT value and also the viral load value, it's just an additional value to be considered. Yeah. And in our hospital, when we consider, because when we find a new F0 in a department, for example, we always have to look at which CT value at the time of the diagnosis. Because if it's very high, I mean above 30, then we feel a bit more easier, right? Because at that point, that F0 is not how to say be able to deliver virus to other people. But when it's very low, like 20 or 16, 17, for example, then we will be
very dangerous,
in very high alert, because we need to look very carefully who have been contacted to that patient, right?
Yes.
Yeah. So the CT value, it's very important at the very beginning, and when we, because our hospital, we also have to establish a new unit of care, COVID care unit in the hospital now, because every hospital have to do that. And then in order to move the patient from that care unit to the other unit, we have to look at the CT value also. So whether the CT value increase above 30, then it is safe to move them out to a separate room outside of the intensive care. Right?
Understand.
Yeah.
Okay, I think this is common in Singapore also, because I think the doctors will use CT value as the semi-quantitative index. Okay, so in the report, they will put the CT value beside the positive result. So then when doctors see it, they can actually be aware of the CT value and manage the patient accordingly. Yeah, I don't think that is that is quite practiced quite widely around the world.
Yeah.
Yeah.
Yeah. Good. Thank you very much.
Yep.
Thank you for very interesting presentation and also data.
Thank you for attending today. Thank you for your time.
Thank
Yes.
you, Chris. Yeah. Các anh chị và các bạn có ai có câu hỏi gì nữa không ạ? Vâng, nếu không thì bây giờ chắc cũng đến cũng hết thời gian rồi, thì xin mời hãng SISC Lan Anh ạ.
Dạ vâng, xin rất là cảm ơn cái phần trao đổi vừa rồi ạ. Xin cảm ơn bác sĩ Quốc Chính ạ. Cảm ơn bác sĩ Thu Hằng. Cảm ơn. Thank you, Chris. Vâng, thưa quý vị, thời gian của buổi hội thảo của chúng tôi hôm nay đến đây là kết thúc. Tất cả các câu hỏi của quý vị đại biểu mà chúng tôi chưa kịp đề cập thì chúng tôi xin phép được gửi email và liên hệ giải đáp trực tiếp với quý vị. Công ty SISC và hãng Thermo xin chân thành cảm ơn sự quan tâm của quý vị dành cho buổi hội thảo ngày hôm nay. Chúng tôi hy vọng với những thông tin và giải pháp hãng Thermo vừa cung cấp, chúng tôi đã đóng góp vào công cuộc phòng chống đại dịch COVID-19 của chúng ta. Xin chân thành cảm ơn hãng Thermo Chúng tôi xin gửi lời cảm ơn chân thành đến các y bác sĩ, các kỹ thuật viên, các chiến sĩ trong tuyến đầu chống dịch đã vất vả hỗ trợ người dân trong công tác phòng chống dịch bệnh thời gian vừa qua. Chúng tôi xin chúc sức khỏe và chúc quý vị đại biểu năm mới 2022 đạt nhiều thành tựu mới để chúng ta cùng nhau chung tay sớm đẩy lùi dịch bệnh COVID-19. Tôi xin chân thành cảm ơn.
Thank you, Chris. Thank you.
Thank you.
Not a problem. Please contact us or SISC for more information if you need help. Okay.
Thank you.
Thank you.
Thank you. Bye bye.
Bye bye.